Okay, welcome to the third bioinformatics and computational biology seminar today. It's a great pleasure today to have Professor Debswapna Bhattacharya. I hope I I pronounced your name okay. So
I don't mind, that's okay.
Deb, okay. So Deb. So Deb has been very successful recently in funding. So he is currently assistant professor in the Department of Computer Science and Software Engineering at Auburn University. And he joined Auburn University in fall 2017. He did PhD in computer science from the University of Missouri, Columbia. And he worked with Professor Jianlin Chen, who is currently one of the successful protein structure prediction team in CASP structure prediction competition. And Deb currently is working on computational biology and machine learning, specifically mostly focusing on protein structure prediction. And recently he got NSF Career Award and also NIH Mira Award. This is a two major funding from both NSF and NIH. So I'm very excited to have him here today. He will be today talking about learning to score and refine protein 3D models. Okay, Deb, please.
Thanks, Daisuke, for the kind introduction, and thank you everyone for joining in. It's great to be back, I would say, to Purdue. I visited once Purdue a couple of years back when I was a PhD student, and was attending one of the summer workshop from people from Japan. It was a great experience. I hope the fact that we are meeting virtually will not sacrifice that experience, but it's great to be back, I would say, to Purdue. Like Daisuke said, that I work in protein 3D modeling, and it's my great pleasure to present some of our latest results in that area. And more specifically, I will talk about learning to score and refine protein 3D models. Let me start by giving a quick intro to the protein folding problem. Actually, the problem is very old and well studied. The statement of the problem is simple. You have the one-dimensional sequence of amino acid, which folds spontaneously into the characteristic three-dimensional structures in order to carry out important biological functions, such as move our muscles, digest food for us, run our immune systems, and so on. So the problem is, how do we understand the process of going from 1D sequence of amino acid to its 3D structure? And more importantly, can we predict the 3D structures given the 1D sequence of amino acid? Despite the simplicity in the problem statement, it turns out the actual problem of protein folding is really hard. In fact, this has been named as 125 open questions by the Science magazine. Here's a quote from the July 2005 issue of Science magazine, which says, out of a near infinitude of possible ways to fold, a protein picks one in just tens of microseconds, and the same task takes 30 years of computer time. So that is the problem itself, when tackled on computer, is computationally very challenging. But this is the quotation from 2005, and a lot has changed since then. Particularly, there's been a dramatic revolution in computing and biology, with biology becoming more and more digital and the integration between computer science and biology. So a natural question is, where are we today? Are we there yet? Can we predict structures of protein efficiently in computer? It turns out this field of protein modeling has attracted a lot of attention recently with the introduction of the AlphaFold method from Google's DeepMind. And this happened in 2018. So in this slide, I have tried to give a random sampling of different news surrounding the AlphaFold method. This one is from Science magazine, which says Google's DeepMind aces protein folding. The Guardian says Google's DeepMind predicts 3D shape of proteins. The Nature magazine says AI protein folding algorithms solve structures faster than ever. Some has gone a step further and argued that this is a watershed moment for protein structure prediction. So in a summary, there is a lot of attention that the field of protein modeling has been gaining due to AlphaFold from Google's DeepMind. So what really happened? Well, AlphaFold participated in the critical assessment of structure prediction, which commonly known as CASP. This is an experiment which happens every 2 years and been happening since 1994. This is kind of considered as an Olympic Games of protein structure modeling. The idea here is research groups all around the world participate in this CASP objective assessment, and then they are asked to predict structures of unknown proteins, which are not necessarily solved by experimental structures yet. And then assessors and evaluators objectively evaluate the progress in the field. The AlphaFold debuted in CASP in its 13th edition in 2018. And this was, in fact, the first group that came from industry side to participate in CASP. It turns out that AlphaFold did very well. Here's a slide documenting its performance. So in the figure, what you see in the x-axis are different groups participating in the CASP 13 structure prediction experiment. And there are around 100 of them. And in the y-axis, it is the overall performance of various methods. AlphaFold actually ranked #1 in CASP 13. And also the difference between the overall performance of AlphaFold and the next best method is quite substantial. In fact, there is a 20-point gap in the sum of z-score, the evaluation metric that CASP organizers use to rank methods between AlphaFold and the next best method from Chang group. So AlphaFold actually did not just rank #1, but it also substantially outperformed the second best method and all other methods. And this is the reason of this, the lot of attention and the news that I just shared surrounding AlphaFold. So what is the reason AlphaFold did so well? Well, it turns out the AlphaFold, instead of trying to predict the 3D structure directly from the 1D sequence of amino acid, AlphaFold breaks the problem down and does it in a sort of 3-stage process. So it predicts the 2D distance map, which is really a 2-dimensional projection of the 3D structure from the 1D sequence of amino acid. And then it tries to reconstruct the structure of 3D molecule of protein from the 2D distance map. And this 2D distance map is really a symmetric real value matrix which encapsulates the pairwise distances between different amino acids. And in particular, AlphaFold uses evolutionary sequence analysis, which has been well studied by Chris Sanders and Deborah Marks' group, and many others over the years. And latest development in deep learning technologies, mostly from vision. Jinbo Zhu's group pioneered this approach, and some other groups also are actively working in this area. But the contribution of AlphaFold is going from 1D to predict the 2D distance map in more finer grain detail and rather accurately in order to reconstruct the 3D shape from the 2D distance maps. It should be noted here, like I said, that AlphaFold is not the only method or even not the first method which tackles the problem this way. Jinbo Zhu's group from Raptor has a method called RaptorX, which works in a very similar way. DMP-Fold from David Jones' group also predicts the 2D distance maps. So AlphaFold really stood on the shoulders of giants and really perfected the recipe for doing that. And this is one of the reasons of their improved performance in this problem and the news surrounding it. But it turns out, due to a lot of buzz around AlphaFold, popular internet is getting flooded with stories like this. This one is from Nanotemper Technologies, asking the question, is the protein folding mystery close to being solved? I particularly like this image, which is, of course, inspired by origami here. But are we there yet? Is the problem really close to being solved? It turns out it is far from it. To understand this, I did a forensic investigation of the PFAM sequence database to understand the current state of protein sequence modeling, protein structure modeling. This is a snapshot of the PFAM protein family database, which according to its 32.0 release, which was released in September 2018, just after CASP 13 finished in 2018. So in total, there are approximately 17,000 plus protein families out there in the PFAM database, and out of which approximately more than half of that, or nearly 51% of that, have already have available experimental structures, meaning that at least one member from these families have been solved by either X-ray crystallography or NMR spectroscopy or cryo-EM microscopy, and resulting in high resolution structures deposited to protein data bank. But then the other half does not have an experimental structure. Out of that half, 16% of families have detectable homologous templates in HH, detectable via HH search. This corresponds to roughly 2,700 families. So for these roughly accurate structure models can be predicted from the sequence using template-based or comparative modeling. Then there is this evolution, then there is this 9% of additional families, approximately 1,600 plus, for which sequence profile information is available, and the evolutionary sequence profile is rather diverse and deep. And that means the methods such as AlphaFold can accurately predict the distance maps for this part of the families. And so this is the realm of AlphaFold and methods such as like AlphaFold. But then there is also this 24% additional family of PFAM family sequences for which the evolutionary sequence either is very low or does not exist at all. This corresponds to roughly 4,000 plus families. And for these, the accuracy of the methods such as AlphaFold is rather low. And so this is beyond the scope of methods such as AlphaFold currently. So this slide illustrates two key points. First, we see that roughly half of the protein families still doesn't have experimental structures. And it turns out that many of these families, it's really hard to determine their structures using experimental means, either because it's impossible, because some of the proteins are not amenable to experimental means, or maybe because they are too time consuming or expensive, or both. The second point is we see that there is a lot of variability in terms of the information. For example, this 16% of the family for which detectable homologous template is available, we can predict the structures rather accurately, like I said before. On the other hand, for these 24% families, these are where there are no or low evolutionary information, the accuracy of structure modeling is actually very low. So this brings us to two key questions. First of all, for the remaining part of the protein families which does not have experimental structures, and where we currently predict the structures using computational modeling, how do we estimate the quality of a predicted protein model and reliably know whether our prediction can be trusted or not? And the second and related key question is, how do we improve the quality of a less accurate model to make it closer, to bring it closer to more like experimental structures? And collectively, these two questions can help us run towards high quality models for the entire protein families. So in the time that remains, I will try to address these two key questions and show us some of the latest results we have been getting in these two key questions areas. So let me do it sequentially by tackling the first question first. How do we estimate the quality of a predicted protein model? So this is actually known as the protein quality estimation problem. Here is the problem statement. We have the computationally predicted protein model. It might come from homology modeling or methods such as AlphaFold. So we really don't know how much information that went into this prediction of the structure. And we don't have the experimental structure. The problem of model quality estimation is, can we accurately predict how accurate the model is? So this is like estimating the quality of the model. It turns out that machine learning plays a very important role in model quality estimation, because machine learning helps to learn a function that maps from protein feature space to some sort of quality score. And naturally, supervised function approximation plays a major role because we have many structures solved by experimental methods. So we can learn the function by observing what the structures that we already know and try to establish this mapping between features and quality score. Now, in terms of features, these methods of quality estimation use standard methods, which include sequence profile, structural features, or statistical potentials. But many of these, none of the methods use the distance features that AlphaFold has been successfully, and methods such as AlphaFolds have been successfully used in structure prediction. And so distance-based features could further improve the performance of quality estimation. In terms of the function approximator, many different variations have been tried, including methods such as support vector machines, random forest, or deep learning methods such as multi-layer perceptron or 3D convolutional neural networks for predicting the quality of the models. But these methods could further benefit from advanced deep architectures. One such architectures, which it turns out to be used in AlphaFold method as well, is residual learning. In residual learning, instead of trying to predict the output Y from the input X, we predict Y minus X from X, and then we add X to the predicted Y minus X in order to estimate Y. And this Y minus X is called the residual. So this is equivalent to adding a shortcut connection between X and Y, as shown in the figure here. The residual learning really enables the training of very deep neural network by stacking many layers together. And this residual learning can be embraced by quality estimation methods to further improve the performance. Finally, the quality score. Here's a widely used quality score, which are called global distance test, or GDT, which are really superposition-based metric for measuring the accuracy of structure prediction, as represented in this equation here. In this equation, the numerator has quantities N1 through N4, which are number of aligned residues of a model below 1, 2, 4, and 8 Armstrong thresholds with respect to the corresponding residues in the native or experimental structures after the structural superposition. And the denominator has this L, which is the length of the protein. This is the number of residues, number of amino acid residues that the protein has. And correspondingly, and consequently, GDT lies between 0 and 1, with 1 meaning the two structures are perfectly identical. And now, just by looking at this equation, we see the quantities N1 through N4, these are threshold-based quantities. And therefore, we can use binary classifications to model them. And furthermore, since there are 4 of them, we can use an ensemble learning for modeling GDT score. And so we can combine the ensemble of 4 different classifiers, each of which targets one of these 4 thresholds, and then combine them together to predict the quality score, which is the GDT score. And so our contribution in this area is these 3 things that I just discussed. Namely, we introduced the distance-based features using distance histogram alignment. We also employed stacked deep residual neural networks, or ResNets, into the model quality estimation. And then we use ensemble learning by learning 4 deep ResNets, each of which targets at one of these specific thresholds. And the result is QDeep, our new protein model quality estimation method. This actually has been recently published in the ISME 2020. Here's a snapshot of the paper. that was just published, and it's online in bioinformatics. So here's a flow chart of the QDeep method. From the sequence of the protein, we extract the multiple sequence alignment, and we extract the standard features, including sequence versus structure consistency, some energy terms. But then we also integrated distance-based weighted histogram alignment, meaning that we extract the distance map from the structure, and then we predict the distance maps from the sequence and figure out how similar they are. And each of this similarity is fed as a feature. We combine all these different features and feed them into stacked deep ResNet, and each of which, there are 4 of them, each of which focuses on one of the 4 thresholds. The architectures of these deep ResNet classifiers are very standard. It includes convolutional block, batch normalization, then residual blocks stacked together, a flattened layer, a dense layer, followed by a sigmoidal activation function. And ultimately, each of these ResNet classifiers predict the likelihood of every residue of a model, of a predicted model, to be below one of these thresholds. And since there are 4 of them, we can combine them analogous to what has been done in the GDT score in order to estimate quality score, which we call QDeep score. Consequently, QDeep score is a probabilistic equivalent of GDT score, which we predict using our ensemble of deep ResNet classifiers. Now, each of these deep ResNet classifiers have been trained using models from CASP9 and CASP10 combined, and we validated this using a standalone CASP11 set. Overall, there are over 2,000 non-redundant models having more than 300,000 residues for over 200 CASP targets. Let me now show you some results. So we evaluated CASP QD performance in CASP12 and CASP13 data sets and compared with some of the state-of-the-art quality estimation methods. And we looked at the various types of correlations, which measures how similar QD is to the real accuracy score, as well as the loss, which measures how our ability to pick the best model from a pool of alternative models. It turns out from both CASP12 and CASP13, QDeep outperforms the state-of-the-art quality estimation methods in a wide variety of metrics. For example, in terms of the average Pearson correlation, our QDeep method delivers in CASP12 a correlation of 0.74, which is much better than ProQ3D, which delivers a correlation of 0.688. Similarly, our loss in CASP12 is also much lower. And same trend continues in CASP13, although the loss of the meshing method is the best in CASP13. We are not the best in that, but we are better in the other categories. But a more important thing to notice here is all the top performing groups, including QDeep, applies deep learning, including the methods ProQ3D and 3D CNN, which applies 3D convolutional neural network, ProQ4 and things like that. So it turns out that deep learning seems to be making some progress in the quality estimation area. Next, we looked at, so a natural question that to ask is, are there other deep classifiers apart from deep ResNets that we can use to, for this purpose? To answer this, we studied, we used LSTM and CNN using the same training data set that we used in QD, and we compared that head-to-head using with our ResNet classifiers using CASP12 and CASP13 data sets. And it turns out that ResNet consistently outperforms LSTM and CNN across all the quality, all the accuracy measures, demonstrating that deep ResNet is a good architecture, deep architecture for the problem at hand. One of the other things, other desirable properties of any quality estimation method is its ability to distinguish a good versus bad models. And the distinction between good and bad is relatively arbitrary, but we choose a GDT threshold of cutoff of 0.4 to classify a model as good or bad. And we studied how reliably various quality estimation methods can distinguish good versus bad models. And here we show the ROC curves, the area under the ROC curves of various methods, including QDeep. It turns out that QDeep outperforms all the other methods in both CASP 12 and CASP 13 in terms of the area under the curve, demonstrating that it is a good method of choice when it comes to distinguishing between good and bad models. We also investigated whether additional information can help. To study that, we used deep profile information coming out from deep MSA method, which include the metagenomic sequences, and plugged in that information into QDeep. And we observed that the performance of quality estimation of QDeep gets further improved when we plug in metagenomic sequences from the deep MSA method, demonstrating that additional information such as deep multiple sequence alignment further improves the performance. But when we take out the distance features from the QDeep method, the performance actually drops, which indicates that distance information is actually a driving factor behind the improved performance of QDeep. So let me summarize this part of the talk before going to the next segment. So the contribution of the QDeep method is threefold. First of all, we introduced distance features. We also introduced the deep ResNet classifiers and applied ensemble learning to come up with a new quality estimation method called QDeep. It outperforms the state-of-the-art quality estimation methods, and we are trying it out in the blind CASP 14 assessment, which are currently undergoing. And deep ResNet turns out to outperform the other deep neural networks such as LSTM and CNN. But it might be an interesting direction to study it further by trying out some of the models that are getting popular in the language models, such as BERT and things like that, and see whether it can further improve the performance, which we are currently doing. We have observed that additional information such as deep multiple sequence alignment helps in improving the quality estimation performance, and distance information is actually a critical factor. Our method is freely available at GitHub, including the source code and training process and everything. You're welcome to check it out. Here is the URL for our method. Now let me go to the next key question, which is related to the first one, which is how do we improve the quality of a less accurate model? This is actually a more important problem, if not the similar important problem than the first one. Because suppose we figure out one model is not so good or moderately good after using quality estimation method. Of course, we would like to make it better and make it comparable to the experimental quality. So we need to be able to improve the quality of a less accurate model. So this comes under the umbrella of protein structure refinement. And the problem here is to bring less accurate model to near experimental accuracy, as shown in the slide here. And for example, we might be given a starting model here, which might have errors in the loop regions, or it could have misfolded secondary structures. And our goal is to go from the roughly accurate model to a very detailed, high resolution experimental quality model, where all the errors in the loops or misfolded secondary structures get corrected. So structure refinement, therefore, is a journey from moderate to high resolution. Now, there are some good news and bad news in this journey. The good news is the starting structures tend to be really close to the target. And this is due to protein modeling and more methods such as AlphaFold and many others methods which are pushing the accuracy of structure prediction. And so the starting structure can be really close to the target. But of course, this is also due to the expanding sequence and structural databases due to the effort of structural biologists. The bad news is traversing this tiny distance is really very challenging. And this is really due to the limitations of scoring, that is evaluating how good we are without knowing the experimental structure, and also due to inefficiencies of conformational sampling. But we need to keep in mind that a mantra in refinement is no matter what, we should not ruin the starting structure, because that is the structure we started with. And if anything, we would like to make it better, not to ruin it and drive it away from the target. And so it turns out the popular refinement methods, when we try to refine structures using either MD simulations or Monte Carlo based methods, and we don't have any restraints for the refinement, we perform unrestrained conformational sampling, then the refinement simulation sometimes drive away from the experimental structures rather than going towards it. And longer sampling does not necessarily help here. So popular refinement method, what it does, it applies restraints which are derived from the starting structures for constraining the conformational space. These restraints are typically applied on the C alpha atoms and using soft harmonic potentials on each C alpha atoms. The idea as shown in the figure here is to restrain somehow the conformational space by applying these constraints on the structures in order to not move away too far away from the starting structure. Now, of course, this leads to a trade-off. If the restraint is very strong, the conformational space becomes very limited, so we get less degree of refinement, but often this leads to improved consistency. On the other hand, if the restraint is very weak, then the enhanced conformational sampling, the possibility of enhanced conformational sampling gets somewhat limited, and we get a less consistency as a result. But it sometimes can lead to improved degree of refinement. So the trade-off is really between the degree of refinement and the consistency. We ask the questions that is, instead of using restraint and deriving restraint from the starting structure, can we predict the restraints using deep learning? And more importantly, can we use multi-scale restraints from the deep learning? We have been able to publish in a paper in bioinformatics. This was published in 2019. This predicts restraints using deep learning and then apply restraint relaxation for structure refinement. The method is called refind-D. Here's the basic idea behind refind-D. So we use standard features derived from the starting structures, which include sequence profile, structural features, and some energy terms. Then we used a classifier, which is a deep CNF classifier. This is due to Jimbo Zhu's group. The deep CNF is really an integration between convolutional neural network and conditional random field. And we used an ensemble of 4 classifiers, similar to QDeep, but since we are doing refinement, we used different thresholds, 0.5 Armstrong, 1 Armstrong, 2 Armstrong, and 4 Armstrong. These are just equivalent thresholds like I discussed before, but focused more on high resolution structures. And again, similar to QDeep, we try to predict the error of every residue to be below one of these 4 thresholds using these classifiers. And so once we feed the features from the starting structures, these deep ensemble classifiers predict the error resolutions at these 4 different error thresholds. And then we convert these errors into multi-scale probabilistic restraints by applying this error on every C alpha atom to be below each of these thresholds. And so these can be converted into probabilistic restraints. So instead of using restraints which are computed from starting structures, here we can use predicted probabilistic restraints. And so each of these residue level local error estimator can guide the refinement simulations. We are calling them multi-scale restraints. And once we guide that, we call it restrained refinement. Each of these classifiers, again, are trained on 2000 non-redundant high-resolution structures, and we optimize the hyperparameters using an independent validation set. Here's the overall flowchart of the refind-D restrained refinement method. Again, we predict the error restraints using the deep CNF ensemble of deep CNF classifiers. which predicts the likelihood of every residue to be below one of these thresholds along with their likelihood values. And then we convert them into flat harmonic restraints and feed them into Rosetta's fast relax method to perform restrained relaxation to generate a pool of refined structures. And then we can combine the error classifications in a weighted fashion, similar to what I just shown in the QDeep method, to score the refined structures and select the best one from the pool. And that is the refined method. That is the flowchart of the refined method, really. Let me show you some results that we got. The first thing we tried is to see the effect of these different multi-scale restraints. So using a data set of 150 non-redundant refinement structures, we studied what if we apply each of these restraints separately. That is, we apply 0.5 Armstrong error restraints, 1 Armstrong, 2 Armstrong, and 4 Armstrong. The figures that you see here, the x-axis represents the delta GDT HA score, which represents the amount of change compared to the starting structure. The green represents the refinement went into the right direction. That is, we went toward the experimental structure. The red or reddish areas represents that we actually ruined the starting structures. And these are all probably the distributions of the different multi-scale restraints. We can see when the restraint resolution is very low, that is 0.5 Armstrong, we more consistently improve the starting structures. So this corresponds to more like 60% or above improvement. On the other hand, when we apply 4 Armstrong restraints, we actually ruin the starting structures more than we improve it. But it also should be noticed here that when we apply 4 Armstrong restraints, the likelihood of getting a high degree of refinement is higher. On the other hand, when we apply very rigid or restraints like 0.5 Armstrong, the likelihood of going likelihood of refinement of more than 5 GDT score points is very low. So therefore, the point here is the restraint resolution can be controlled to achieve the trade-off between degree and consistency on demand. That is, we can control, if we want consistency, we can apply the more rigid restraints. On the other hand, if we want more degree of variability and degree of refinement, then we can apply more softer restraints. To try that out, we actually came up with 2 variants of refined method. One in which we applied all these restraints cumulatively. That is, we put all of them together and applied them simultaneously. And then in the other version, which we call non-cumulative version, we applied each of these restraints one by one and generated a pull-up model and scored this pull-up model using the scoring function I just described. So this resulted in 2 flavors of refined. One is refined cumulative and the other is refined non-cumulative. And we compared the performance of that using the 150 non-redundant single domain proteins where the starting structures are predicted using ITESR models. And then we compared refined both the cumulative and non-cumulative versions with several refinement methods, including the molecular dynamics simulation method, FGMD, Rosetta's fast relax protocol, and many different variations of the fast relax protocol, the mod refiner method, which also is energy minimization method. And we looked at the average delta, average score change of the top 1 and the best of top 5. It turns out when we look at the best of the top 1, refined cumulative leads to a more consistent performance. And it in fact delivers the best average delta compared to all other methods, and it's statistically significant. On the other hand, if we go to non-cumulative version and look at the best of top 5, it delivers actually much better performance, and the average amount of delta is also much higher, but it is not necessarily very consistent, as can be seen by the performance of the top 1 average delta of top 1. So the bottom line is refined C or refined conservative leads to a conservative refinement mode, and refined non-cumulative is an adventurous refinement. And depending on our scenario and what we want, we can try to use either conservative mode or non-conservative. For example, if our scoring function predicts our structure to be already very good and close to native, maybe we can try conservative refinement. On the other hand, if the structure is far away from native, we may try the adventurous mode. We can also combine these modes together. We can try adventurous mode first and then try conservative mode afterwards in a sort of cascaded manner. We are actually experimenting with some of these cascading of different modes currently. This is a kind of a case study of what refined non-cumulative mode can do and how much improvement that it can lead to. These are, of course, good examples. Here, the starting structure is in orange, the refined structure is in blue, and the experimental structure is in green, all superposed together. And we can see for some of the cases, for example, this one, the amount of change is actually quite high, more close to 30% delta GDT-HA improvement, for example, from 53 GDT score to 69 GDT score, which represents substantial structural improvement. And also some of the other examples, we see the similar trend. So refined can make pronounced structural improvement when we use the non-cumulative version, but we should be also mindful that it is not always the case. These are all good examples. Sometimes it is not the most consistent method. Sometimes it can degrade the starting structures. We participated in the CASP 13 blind assessment using refined non-cumulative mode, which I just shown here. This was actually officially ranked among the top refinement methods, not necessarily the best method, but among the top. It is interesting to note here that methods which are ranked higher than us are mostly using very computationally expensive empty simulation. By contrast, our method is very, very fast. We just can predict the multi-scale error restraints from using deep learning very quickly, and then we apply Rosetta Fast Relax, which also are very computationally efficient. It turns out the performances, our performance is also comparable to some of the top groups out there in terms of blind refinement assessment. Let me now summarize this part of the talk. In the refined D method, we introduced deep learning-based restraints for improved refinement. And we figured out that restraint resolution can be controlled for achieving degree or consistency on demand. If we care about degree, then we can get substantial refinement, which are as high as 30% accuracy improvement. The method is also ranked among the top refinement method in blind CASP 13 assessment. And also this method is freely available as a web server. You can paste your starting structure and choose what refinement method you want to try, whether you want to try a conservative mode or an adventurous mode. And this web server is going to perform the refinement and return the result back to you. I'd like to speculate the road ahead and then show you some of the directions and briefly discuss some of the directions we have been trying recently. So we have been trying scoring and refinement using deep learning. And more importantly, we have been integrating distance information. But very recently, methods such as TR Rosetta and many other groups have been using orientations in addition to distance information. The orientation, by orientation, I mean how the orientation between the contacts or distances of inter-residues, amino acids of different residues. So it would be interesting to integrate the orientation information in addition to distance information in our methods. For example, QDeep, we can integrate orientation information from methods such as TR Rosetta. But another important point is, in the QDeep method I discussed, we have been not explicitly but implicitly using evolutionary information, because all our profiles, all our features are actually internally using evolutionary sequence profile. It would be interesting to try scoring and refinement without using any evolutionary information, that is, without trying to use any multiple sequence alignment, and just using one single sequence and predict the quality score, or trying to refine model without using any multiple sequence alignment information. This is important because for many of the protein families we have been trying in the PFAM database, it does not necessarily have deep sequence alignment. So we really need to get rid of our dependency on MSAs and come up with a pure ab initio method. We can also try advanced deep architectures. I already mentioned that many deep learning is a very active area, so we can try many different recent developments in deep architectures. And we can also try not necessarily supervised machine learning. We can try something like unsupervised or semi-supervised learning to see whether we can further improve the performance of the quality estimation and refinement, and really in order to improve the structure modeling accuracy and push it towards the high resolution near experimental quality. Speaking of semi-supervised learning, I thought I would conclude by showing you something semi-scientific. So this was a poster that we presented in ISMB Art in Science exhibit in 2020 ISMB. We call it multiple sequence alignment. What we did here, we took a multiple sequence alignment, which are for a protein family, for a particular protein sequence, which are very diverse and have a lot of sequence, is very rich and diverse. And then we perform what we call systemic deletion manually. And this is just to remind us the inequality that exists out there, and this is really a call for equality, so we call it multiple sequence alignment. Finally, I'd like to acknowledge the students, my students, particularly Hussein, who did most of the work in QDeep work. Hussein and Shrutanu also contributed into the Refine New work, and many others. Some of the group members like Muhammad has recently joined. Some others are really getting trained. I'd also like to acknowledge the funding sources, NIH, NSF, and Auburn University for supporting our work. Also Exceed for providing computational resources for our work, and Alabama Supercomputer Authority for providing computational support. And I would also like to thank you for your kind attention. And with that, let me stop here, and I'll be happy to take questions.
Okay, thank you very much for a very interesting talk. Okay, let's see if anyone has question.
Hi, Deb. So I have a question. So I think many years ago, I heard one of the CASP talk from some guy from Michigan State University. And he's working on a model refinement. And I think he mentioned that right now, even for MD methods, so folding from scratch is still very slow. But if you're using MD to actually refine a structure who is already folded, so it's kind of fast. So I think, so my question is really that, so your model, your refinement approach is based on statistical approach and deep learning. And whether the refinement is orthogonal to the MD-based methods or not. Or in another way of speaking, can you first adopt your methods to refine a structure, and later you can observe further improvement by using MD methods? It's kind of like, for sometimes we really don't care about the time actually. So, if we focus one structures, do you think it's possible that first use your methods, then using MD methods to have this two-step of refinement?
That's an excellent suggestion. I think it's very much possible. I think you're referring to the Michael Feig's group from Michigan State.
Yeah, exactly.
Yeah, so I think Michael Feig's group has been publishing some methods where they took the AlphaFold prediction, which has been using deep learning, and then they are trying to use MD simulation to refine it. So the short answer to your question is, of course, I mean, that would be a very interesting direction wherein you do first a coarse grain sort of refinement using a quick statistical sampling approach, and then you do a more detailed full atom resolution with maybe explicit solvent model and things like that. That would be a really interesting direction. I think the Michael Feig's group have been trying to refine some of the COVID-19 proteins,
really?
SARS-2 proteins using their MD simulation. I think so. It is very much possible. The question the one issue that may come up is, if you cascade two methods, which one to do first? Whether you want to do MD simulation first, or you want to actually try coarse grain statistical approach first, because it really also relates back to how accurate your starting structure is and how accurate you can predict it.
Yeah.
If it is accurate, then maybe you can directly do a full atom MD simulation right away.
Right.
On the other hand, if it is not that accurate and you don't have, or you don't have confidence on your quality estimation, maybe a two-step or maybe a multi-step approach a good idea.
Right. I see. Yeah. I got it. Thanks.
Okay. Any other questions?
I have a question. Hi, Deb. I'm Depan, as postdoc in Professor Chiara's lab.
Hi.
So about the refinement, the comparison you showed in one of your slides. So I see that you're comparing the GDT, which is one which has like about 43% of the weight in the score. But if you go to one of the slides there, yeah, exactly this slide. Yeah, the next one.
This
the
one
next
or
the next, the structure.
Okay, the structures. Okay.
Yeah. So now, if we look at the left panel, right, you see that the green loop region over there, which is the experimental structure, right? That the bottom left, please.
Okay. This one.
Yeah. Yes. So you see that even the GDT is so high, but in RMSG it will make a very bad penalty.
Yeah.
So how do we deal with such things?
Yeah. RMSD is very sensitive to errors in the loop regions or subtle errors. So you're right. And of course, if I were to evaluate the RMSD, in fact, in the paper we show the RMSD of this method. I'm not showing it here, but in terms of RMSD also, the refinement is improved. But you need to keep in mind two things. First of all, RMSD is not necessarily a normalized metric. It's not length normalized. So you don't necessarily you cannot compare different sized proteins using RMSD. And the second thing is this is terminal region, so it might be a little more flexible than the core region of the protein. So I would say this matters less that whether we predict the terminal region more accurately than whether we are able to predict the core region or move the core region towards the native structure. But yes, you're right. There is quite a lot of variability in the loop region here. And RMSD will show it, but whether to use RMSD is another question.
Right. And the other question was in the protocol you mentioned I had for the refined D protocol. Yeah, so in this case, the flat harmonic part is from Feig, right? Like he proposed that one. But my question is this: like, when you use this flat harmonic, is this flat harmonic actually part of the Rosetta pipeline, or how is it? It's not very clear to me that how atoms are actually moving. Like what?
Okay.
Yeah, like
in
the deep learning pipeline. Yeah.
Sure. So the contribution of deep learning is really to predict the probabilistic restraint. So, and after that, we use a very standard Rosetta fast relax. So the actual sampling is really dealt by Rosetta fast relax. And so what we did is, going back to your question on the flat harmonic, this is actually a standard score function in the Rosetta suite. So what we did is we plugged in the restraints that are predicted from deep learning into the Rosetta scoring function. So we use the full atom Rosetta scoring function, plugged in these flat harmonic restraints into the scoring function, but then we also weighted it using the likelihoods that are predicted from deep learning. And so what is guiding the fast relax is really a combination of Rosetta's energy function and the classification that are predicted from our deep learning model. And so there's, under the hood, what is happening in the restrained relaxation is really fast relax running with the flat harmonic restraints, which are predicted from deep learning.
Okay. Thank you. Yeah.
Yeah, sure.
Any other questions?
I have questions.
Sure.
My name is Genki. So
Hi,
I have
Genki.
questions. Hi. I have a questions about the refinement method. So what is the most important information to do the good refinement? For example, the accuracy of the contact predictions or number of the multiple sequence element or initial quality of the initial structures. What is the most important factors?
That's a really good question, Genki. We studied that. It's one thing is absolutely clear: the starting structure, the quality of the starting structure is absolutely very critical. If the quality of the starting structure is not very good, most often than not, we actually damage the structure when we try to do refinement. When the starting structures are good, by good I mean when let's say the TM score is more than 0.5 or 0.6, or maybe even approaching 0.7 margin, then we see that not just our refinement, but the other methods that we try to compare our method against, they all tend to do very well when we have a reasonably accurate starting structure. When, on the other hand, we have, let's say, a model which are not necessarily very good, then we have seen that almost all methods actually have a tendency to deteriorate the structures. Going back to your question, and I think this question really goes back to the first question that is being asked. So going back to your question, which feature is more important? We studied that. We have not included that in the manuscript, but it's not entirely clear that whether these energy terms or structural features are more important, but sequence alignment actually is very important. Particularly the depth of the MSA matters, but we have not seen a dramatic effect. Therefore, we have not included in the paper. But when we take out one of the features, we see the classification performance drops, but we have not been able to actually study whether that actually is directly correlated to the refinement performance, because things become clumsy when we perform restrained relaxation using the restraints. It's not very straightforward to objectively judge which features are driving factor behind refinement. But the depth of the MSA matters, and the quality of the starting structure plays a very important.
Thank you. Yeah.
Okay, other questions? Okay. Well, I have, okay, some questions. So here in this, well, okay, let's start from this refined D. So in this refined D, the equation R is either 0.5, 1, or 2, or 4, I guess. And then you combine all of them in summation. So, but in QDeep, you also have a prediction of residue level error at the individual. Okay, again, in the steps. And is this okay? Actually, if it's a prediction of refined D, I think it's similar or correlated at least from the prediction you have from QDeep. And what about just using the QDeep prediction directly put into the this refinement protocol?
Yeah, that's a good observation, Daisuke. So we actually did the refined D work first. So we started with the deep CNF method to predict the performance, to build the classifiers, and then we actually went on and did the QDeep work. Oh. It turns out the QDeep classifiers are more accurate than this work, and this is because probably because we maybe due to ResNet architectures, use of ResNet architectures, and maybe more engineering, hyperparameter optimizations and things like that. And so you are right, we actually can predict these restraints directly from the similar architecture that we have discussed in QD. In fact, that's the method we are testing in the CASP 14 assessment currently, where we are currently integrating the QD based likelihood estimator and the same architecture. So we replace deep CNF classifiers with the ResNet classifiers, because we see that is giving us the better performance. And it also improved. We actually we are actually writing out a paper on that, where we compare this version of the refind-D with the newer refind-D method. It turns out when we use the ResNet classifiers, the performance is a little bit better when we combine the restraints together. Yeah.
Okay. We can take probably just one more question. Anyone has any questions?
I have a small question.
All right, all right.
Thank you, Professor, for the interesting talk. How feasible do you think it would be to extend QD and refind-D to multiple protein chains?
Oh, that's a very good question. Currently, it is not possible. Currently, it can only handle a single chain. The QD method it can be extended for multiple chains, provided we can get the multiple sequence alignment information for the multi-chain protein, and we can reliably estimate the interchain contacts or distance predictions. If we can do that, then it can be extended. Because the architecture is neutral, we can easily extend that architecture for complex prediction or complex model quality estimation. The refinement method is rather tricky, because here we are talking about applying restraints between chains and actually sampling the structures. So although Rosetta suite of method can handle multi-chain proteins, we have not tried it. So if I were to guess, I would say it requires probably a separate study to see the feasibility. So, we have not tried it. Currently, the methods can only handle multi-chain. It would be an interesting direction, actually, so to try it on multi-chain. So thanks for the question.
Okay. So with that, we will thank you. We thank the Professor Bhattacharya again for a very interesting talk on the state-of-the-art protein structure prediction methods. Thank you very much.
Thanks, Deb.
Yeah.
Thank you. Thank you so much. It was great to be here, and I hope that I can come back physically to Purdue once
sure. Yeah.
situation.
Yeah. If you have time, I will invite you many times in the next 5-10 years, or maybe next year when the COVID-19 stuff is done.
Yeah.
Okay.
Okay. Nice to see you.
Yeah. Thank
you.
Okay.
Really nice to meet you here.
Thank you.
Thank you.
Thank you very much. So, okay, so just a next announcement of the next seminar. Next seminar will be on 2 weeks from now, October 22nd, by Professor E.G. Wan from Indiana University. And the talk is about building gene regulatory network at single cell level. This is another very interesting, hot topic. So please join us again. Thank you very much, Professor Bhattacharya again, and then thank you for joining us.
Thank you.
Well, thank you guys. Bye. See you next time. Bye.
Bye.